infidelity. It cuts round about the target area, which promotes you can attach a reporter system to it, a colorimetric or fluorometric reporter system. So here, what they did is that they combined double-stranded DNA and RNA together using RPA amplification or recombinase polymerase amplification. Has anyone worked with RPA before? This is a relatively new technique to amplify nucleic acids, and it's an isothermal approach, meaning it only needs one temperature. So you can actually put all your reagents in a tube and hold it with your hand and reach it between 37 and 42 degrees, and within a few minutes you would have ample amplification. So it is a very powerful tool. It's a new tool. I don't really understand the mechanism yet of it, but it is quite interesting, and I'm excited to look into this further. So you have the target gene in blue. So you transcript using T7 transcription once amplified, and then you expose it to the Cas13a system, and if it cuts, it will report. So here is the amplification, or excuse me, the detection using Cas13 alone, and coupling it with RPA amplification. You can get up to 1 attomole of sensitivity, and that even beats the hell out of PCR. So you have 1 attomole of sensitivity. So here's the, as I said, usually CRISPR will cut specifically at the target. Here it is quite promiscuous. It will cut upstream and downstream, allowing attachment of reporter molecules. So they've used it for a plethora of different approaches. Detecting the presence of Zika virus in blood and urine samples of patients very quickly. Distinguishing between the genetics of same species, or for example, African and American Zika virus quite rapidly. Detecting antibiotic resistance, and most importantly, identifying cancerous mutations in simulated cell-free DNA. And this is the great thing, that you can actually take a sample from a patient in the field and get the paper form of the CRISPR-Cas13a system is not as accurate as the liquid form, but you can get up to 20 attomole accuracy from localized paper-spotted system. I'm not sure if you can see it here, but as I said, they've used the method to detect a recombinant, very small amounts of Zika virus within a lentivirus construct. So they have small sequences of Zika virus, and they saw if they could identify it. They could. They also did a Zika and dengue recombination. So they have one virus that has aspects of Zika and dengue virus hidden within their genome, and they had the ability to detect it, both of them simultaneously. So you cannot cheat it. That is really, really amazing. Single base pair annotations and single base pair changes are very easily detected down to, as I said, an attomole of sample. So mismatches and single nucleotide changes can be detected rapidly. Also, you can identify human SNPs quite efficiently and very cheaply. So here's a diagram of the human genome, and at one run, different SNPs for individuals are identified almost instantaneously. So the advantage of the CRISPR diagnostic system is it can be paper-based. So actually, it can be localized and spotted on paper and sent to places where you have infectious diseases or outbreaks of viral diseases. It is a point-of-care, as I said, diagnostic tool because of its versatility and utility. Cheap. $0.50 for each run, for each test on paper. So it is that cheap. This ability to take raw samples and immediately start processing could transform the diagnosis of Zika and boundless number of other infectious diseases. So this statement has been done by a leading leading person in the field of combating infectious diseases. So the future. I intended to give this talk to encourage the adaptation of this technology. This technology is now available commercially. You can use it for your different research. You can play with it. You can understand it while it's cheap. Because there's a war going on between the Broad Institute in Harvard and MIT and Professor Doudna at Berkeley. There's a patent war going on. And this will be resolved very soon. And once it's resolved, the prices will skyrocket. You'll have to pay a generous amount of money for each one of these one of these people. So I think it is a good time to understand this technology, to adapt it in our labs, to really look into it, because as I said previously, anybody with very minute basic skills in molecular biology and genomics can adapt the system very easily. The programmability is based upon construct delivery and transposon sciences, which is quite a straightforward science in molecular biology, and therefore there's a gold opportunity to adapt this, as I said. So with this, I end my talk, and I thank you very much for not sleeping during my talk. Thank you.
Thank you, Dr. Yazid. I think we're going to have also the question at the end of this session. Now we're going to have Dr. Hussam. Hussam Zawawi. He's an assistant professor of clinical microbiology at the College of Medicine, King Saud bin Abdulaziz University of the Health Science. He developed a track record in surveillance, diagnosis, and awareness of the antimicrobial resistant. He was recognized by receiving multiple national and international awards, including King Abdullah International Medical Research Center. Welcome, Dr. Hussam.
Thank you so much. Before I start my talk, I would like to express my gratitude for this very nice invitation from Dr. Salman and for taking the place of Dr. Fatima Al-Hamlan. I highly appreciate the space she generously given. My talk will be actually just tapping on the aspect of using proteomics for the sake of improving diagnosis, particularly against antimicrobial resistance. Fox hunting